All right, let's talk about a global health challenge that honestly doesn't get nearly enough attention, but affects billions of people. This is more than just a story about parasites. It's a full-blown evolutionary arms race. You've got human ingenuity on one side and microbial adaptation on the other. For anyone studying microbiology, understanding this fight against parasitic worms or helminths is like getting a masterclass in survival and resistance. So let's jump right in.
So here's our game plan. We'll start by looking at the sheer scale of this hidden pandemic. Then we'll properly meet the culprits, the helminths themselves. After that, we'll check out the drugs in our current arsenal, see how they work. And then, the crucial part, how the enemy is evolving to fight back. We'll wrap up by looking at the cutting edge of research, which is where the future of this whole battle is being decided.
Okay, part 1, a world of worms.
Let's just start with the scale, because the numbers here are absolutely staggering. This map lays out the global distribution of what we call soil-transmitted helminths. These things are one of the most widespread infectious agents on the entire planet. As you can see, the highest prevalence, all that red you see, is concentrated in tropical and subtropical regions.
And that brings us to this number, 1.5 billion. Of the over 5 billion people considered at risk around the globe, it's estimated that 1.5 billion are infected right now with at least one type of these worms. That's nearly 1 in 5 people on Earth. Just let that sink in for a moment.
So, who exactly are these antagonists? It's time to meet the major players in this story.
The term helminth basically just refers to worm-like parasites. Now, from a clinical point of view, we classify them based on 2 key things. First, their morphology, their external shape, and second, which of the host's organs they decide to call home. This chart breaks it down nicely. It all boils down to 2 major phyla. On one side, you've got the nematodes, which you probably know as roundworms, and on the other, the platyhelminths, or flatworms. Now, those flatworms are then split into other classes, like cestodes, those are your tapeworms, and nematodes, the flukes. And here you can see that fundamental difference perfectly. Look at the left. That's the classic cylindrical shape of a nematode. Now compare that to the trematode on the right. It's totally different, right? Visibly flattened, it almost looks like a leaf. This basic round versus flat idea is really the first step in telling them apart. So we've got a massive global problem, and we know who the enemy is. So how do we fight back? Well, let's take a look at our current anti-worm arsenal. For decades, really, our frontline defense has relied on just a handful of these core drugs. Names like albendazole and mebendazole are the absolute workhorses for most soil-transmitted helminths. And then you have something more specific, like triclabendazole, which is the go-to drug for liver flukes. So how do they actually work? Well, the genius of drugs like albendazole is their specificity. They're designed to selectively target and block the polymerization of something called beta-tubulin. By doing that, they stop microtubulins from forming. And this is a huge deal because it basically dismantles the parasite's cytoskeleton, a structure that's absolutely vital for things like nutrient uptake and cell division. This mechanism is essentially a 2-pronged attack. First, by knocking out those microtubules, the drug stops the worm from absorbing glucose. It literally starves it. At the exact same time, it grinds mitosis to a halt, which stops any egg production. So the parasite is just left there, immobilized, unable to feed or reproduce, and eventually, the host's body clears it out. But here's the twist. While we've been relying on this same arsenal for years, the enemy has been busy. It's been learning, adapting, and evolving. And this is where the story gets really challenging. This quote really says it all. We've relied on a tiny number of drugs for so long, especially triclabendazole for liver flukes in livestock. This has created an immense selective pressure. The first resistant strains popped up back in 1995, and what used to be seen as just a veterinary problem is now a really serious concern for human medicine. So, how are they doing it? How are they developing this resistance? Well, the scientific community has landed on 3 main models to explain it. Let's break them down one by one. The first one is the most intuitive, an altered drug target. Remember how the drug is designed to bind to that beta-tubulin protein? Well, imagine a mutation occurs that changes the shape of the protein's binding site. Suddenly, the drug can no longer lock on. It's like the parasite has changed the locks, and our key just doesn't work anymore. And this really highlights a major gap in our knowledge right now. We can see the end result, the phenotype. We can see microtubule disruption in sensitive worms, but not in the resistant ones. But the precise molecular reason, the genotype, is still a bit of a mystery. And the big challenge here is that resistance doesn't seem to be from a single simple mutation. It's probably polygenic, meaning it involved multiple genes working together, which makes finding the exact cause incredibly difficult.
This challenge, though, this is exactly where the next generation of microbiology research comes in. So let's look ahead to the next frontier and what the future of this fight might look like. The strategy going forward has to be a multi-pronged attack. There's no single silver bullet here. We're looking at everything from repurposing drugs we already have to using clever drug combinations to avoid the problem of using just one drug. We're also developing new inhibitors, screening natural products, and of course, using all the amazing advances in genomics and proteomics, the whole omics field, to find new biomarkers.
This diagram shows you the new battleground, the parasite's own cellular machinery. Instead of just trying to blow up its cytoskeleton, what if we could sabotage its defense mechanisms? For example, we're looking at inhibiting these efflux pumps, like these multi-drug resistance transporters you see here, that the parasite literally uses to pump our drugs right back out of its system. If we can block those pumps, we might be able to make resistant worms sensitive to our old drugs again. And this leads us to what is, for me, one of the most exciting new frontiers, the microbiome. Researchers are seriously exploring whether the host's own gut bacteria could be a source of brand new anti-worm compounds. Just imagine if we could find bacteria whose natural byproducts are toxic to worms or that tweak the host's immune system to fight the infection better. It's a total paradigm shift.
Which brings us to our final thought. We are in a constant, high-stakes evolutionary arms race. As these parasites continue to evolve, our science has to evolve even faster. For all of you, as the next generation of microbiologists, the next massive breakthrough in this global fight could very well come from your work. The only question is, where are you going to look first?